Good morning, my name is Pawandeep and I'm going to explain you the structure of DNA. So DNA basically is deoxyribonucleic acid and it is a genetic material in most of the organisms. Before studying DNA structure, we must know about nucleosides, nucleotides, nitrogenous bases and nucleic acids. So nucleosides are the combination of nitrogenous bases, pentose and pentose sugar. Nitrogenous bases are the bases containing nitrogen and pent means 5. So the sugar that has 5 sides or 5 carbons is known as pentose sugar. Example of such sugar is ribose and deoxyribose sugar. So this is basically the structure of pentose sugar, which is a typical pentagon and it has carbon arrangement in such a manner. Here the first carbon is present, the second is here, third is here, fourth is here and fifth carbon is present here. And this particular position is occupied by a single oxygen atom. The next thing is nucleotide. Nucleotides are the combination of nitrogenous bases, pentose sugar and phosphate. So nucleotides are the combination of nucleosides and phosphate. So when nucleosides and phosphate combine, it forms nucleotides. Now nitrogenous bases, they are of two types, purines and pyrimidines. Purines contain nitrogen at 1, 3, 7 and 9 position. Pyrimidines contain nitrogen at 1 and 3 position. The rest position in both of the nitrogen bases are occupied by carbon. The structure of purines is that of a hexagon attached with a pentagon, and structure of pyrimidines is that of a hexagon. Examples of purines are adenine and guanine, and pyrimidines example is cytosine, uracil and thymine. So what is basically deoxyribose sugar? This is the structure of ribose sugar. At second primer position, we contain an OH group, and when the oxygen is removed from here, it forms deoxyribose sugar. So what DNA is basically, it is the combination of deoxyribose sugar and nitrogenous bases. The nitrogenous bases are adenine, guanine, cytosine and thymine and phosphate. So this forms the single unit or single nucleotide of DNA. When certain nucleotide combines, it forms a polymer of deoxyribonucleotides, and the polymer of nucleotide is referred to as nucleic acid. So this is basically deoxyribonucleic acid or DNA. That means polymers of these nucleotides forms DNA. Now what is the length of DNA? Length of DNA can be defined as number of nucleotides or number of pair of nucleotides. न्यूक्लियोटाइड्स का कितना नंबर है और न्यूक्लियोटाइड्स के पेयर का कितना नंबर है, और न्यूक्लियोटाइड्स के पेयर के नंबर को हम बेस पेयर बोलते हैं. So if we take the single strand of DNA, so if suppose it contains X nucleotides or X nitrogen bases, so its length would be X nitrogen bases or X nucleotide long. Now when it will pair with the complementary strand, so the nitrogenous bases present here will attach with each other, and thus it will form paired nitrogen bases or base pair. That is why for double stranded DNA or two strands when combined, we say that it is X base pair long. Suppose the value of X is 10. So this forms 10 nucleotides long DNA and this forms 10 base pair long DNA. So now what is complementary strand? So complementary strand का मतलब ये होता है कि when this strand will bind to the other strand of the same kind, it should be complementary to each other. So complementary कौन हो सकता है? देखो इसमें भी नाइट्रोजन बेसिस प्रेजेंट है, इसमें पेंटोज शुगर प्रेजेंट है और फॉस्फेट प्रेजेंट है. पेंटोज शुगर और फॉस्फेट में तो कोई डिफरेंस है नहीं. जो डिफरेंस हो सकता है वो नाइट्रोजन बेसिस में हो सकता है क्योंकि उसी में वैरायटी है. So if suppose this strand contains some else nitrogen bases and this strand contains some else nitrogen bases. So what complementary principle tells us about is that adenine always binds with thymine and cytosine always binds with guanine in a DNA structure, and adenine binds with thymine with double bonds and cytosine binds with guanine with triple bonds, and these bonds are hydrogen bonds. In DNA the case is, in RNA the case is that the thymine is replaced by uracil and rest remains the same. So if suppose this strand of DNA contains adenine, guanine, cytosine and thymine here. So this strand must contain, this black one strand must contain these bases as thymine, cytosine, guanine and this one base would be of adenine. So that is why then only the pairing will occur, else this strand won't pair with this strand. Now base pairing, the base pairs present in DNA is adenine, thymine, cytosine, guanine, and in RNA thymine is replaced by uracil, which causes a difference of DNA and RNA. Now examples of length of DNA. E. coli contains 4.6 into 10 ki power 6 base pair long DNA, and haploid human DNA contains 3.3 into 10 ki power 9 base pairs long DNA. Now what is a structure of polynucleotide chain? Polynucleotide means number of nucleotides, and number of nucleotides form nucleic acids. So we are going to study the structure of nucleic acids. The number one is nucleosides. First the nucleosides formation occurs. So how this occurs? When ribose sugar, we are talking about DNA, when the ribose sugar, deoxyribose sugar actually binds with the nitrogen base through N-glycosidic linkage, this forms nucleosides. Now, the next structure formed is nucleotide. So when this structure, when nucleosides, that means nitrogen bases plus ribose sugar के साथ pentose जुड़ जाता है, sorry phosphate group जुड़ जाता है, so it is joined by phosphoester linkage, and this is joined with the fifth carbon. So this forms a nucleotide, and phosphate is joined in the form of this particular thing. Here it is phosphate double bond O. This end is bounded to the carbon. This oxygen is bounded to the carbon. This is oxygen minus oxygen ion, and this is also oxygen ion. So when number of nucleotides, such nucleotides bind, they form a polymer. So this forms a single strand of DNA, and these nucleotides are linked by phosphodiester bond. And when the nucleotides are linked, the product released is water. So this forms one strand, and then pairing occurs, which is complementary base pairings, and this forms DNA. So this particular structure of DNA is known as double helix structure, and it was given by Wilkins and Franklin. Now talking about how we give the names to nucleosides. So if suppose the sugar is ribose sugar and the base is adenine, so the name would be adenosine, and the sugar becomes deoxyribose, so it will become deoxyadenosine. Same is the case with guanine and rest of the nitrogenous bases, guanosine, deoxyguanosine and so on. Now one important thing is there, the ribose binds with nitrogenous bases at sites that has nitrogen on first, which is the lowest, and ninth position, which is the highest, and highest position is preferred. इसका मतलब ये कि जब राइबोस शुगर नाइट्रोजन बेसिस के साथ पेयर अप करेगी, वो सिर्फ दो पोजीशन पे पेयर अप करती है, जिन दो पोजीशंस पे नाइट्रोजन होता है. सबसे पहले वो नाइन्थ पोजीशन के साथ पेयर करती है, जिस नाइन्थ पोजीशन पर नाइट्रोजन होगा उसके साथ पेयर करेगी, या फिर फर्स्ट पोजीशन पर जहां पर नाइट्रोजन होगा उसके साथ पेयर करेगी. एंड जो हाईएस्ट पोजीशन है उसे हम प्रेफरेंस देते हैं. सपोज प्यूरीन्स में नाइन्थ पोजीशन पे वाला जो नाइट्रोजन है वो इस राइबोस के साथ बाइंड करेगा, एंड पायरिमिडीन्स में हाईएस्ट तो है नहीं, सो वी हैव टू टेक द लोएस्ट वन. सो हम 1 वाला जो नाइट्रोजन होगा वो इस राइबोस के साथ बाइंड करेगा. नाउ नेक्स्ट आर द कंक्लूजन्स. सो ये स्ट्रक्चर जो बना, इसकी वजह से हमें क्या पता चला कि एक एंड पे फ्री फॉस्फेट प्रेजेंट होता है और दूसरे एंड पे फ्री OH प्रेजेंट होता है, जिसका मतलब हमें दो एंड्स मिल गए. सो जिस एंड पे फ्री फॉस्फेट प्रेजेंट होता है उसे हम 5 प्राइम एंड बोलते हैं, जिस एंड पे फ्री OH प्रेजेंट होता है उसे हम 3 प्राइम एंड बोलते हैं. The next is backbone of polynucleotide is formed by sugar and phosphate. See, what is backbone? It provides structural framework. So structural framework of DNA is provided by sugar and phosphate. See, sugar and phosphates are present at this site. This strands are made up of sugar and phosphate. But the nitrogenous bases, they are protruding inwards. तो वो framework बाहर वाला नहीं बना सकते. That is why हम नाइट्रोजनस बेसिस को बैकबोन में इंक्लूड नहीं करते हैं, जबकि नाइट्रोजन बेसिस जो होते हैं वो भी स्टेबिलिटी प्रोवाइड करते हैं हाइड्रोजन बॉन्ड्स बनाने की वजह से. एंड वन मोर थिंग, एंटी पैरेलल स्ट्रैंड्स. द स्ट्रैंड्स आर ऑलवेज एंटी पैरेलल. सपोज this is the 5 prime end of DNA and this is the 3 prime end of DNA strand, and this is the 5 prime, this is the 3 prime. So 5 prime end won't bind with 5 prime end, so this thing won't occur, this won't occur, and that means the strands are always anti parallel, against parallel. Parallel का उल्टा, मतलब ये ऐसे bind करेंगे 5 prime with 3 prime and 3 prime with 5 prime. This won't occur and this will make the structure of DNA, and this is the straight structure of DNA. Nitrogen bases are present here and this is the coil. So this way our DNA is formed.